Signal transduction. Cell signaling can be divided into three stages. Reception: a cell detects a signaling molecule from the outside of the cell. A signal is detected when the chemical signal, also known as a ligand, binds to a receptor protein on the surface of the cell or inside the cell. Transduction is the second step. When the signaling molecule binds the receptor, it changes the receptor protein in some way. This change initiates the process of transduction. Signal transduction is usually a pathway of several steps. Each relay molecule in the signal transduction pathway changes the next molecule in the pathway. Response: finally, the signal triggers a specific cellular response. Soluble receptors. Ligands. The majority of signal transduction pathways involve the binding of signaling molecules known as ligands to receptors that trigger events inside the cell. Steroid hormone receptors are found in the nucleus, cytosol, and also in the plasma membrane of target cells. They are generally intracellular receptors, typically cytoplasmic or nuclear, and initiate signal transduction for steroid hormones, which lead to changes in gene expression over a time period of hours to days. Vitamin D hormone binds to receptors in its target cells, controlling the synthesis of many different proteins involved in calcium transport and utilization. The prostaglandins are a group of lipids made at sites of tissue damage or infection that are involved in dealing with injury and illness. They control processes such as inflammation, blood flow, and formation of blood clots, and the induction of labor. Thyroidothyronine, also known as T3, is a thyroid hormone. It affects almost every physiological process in the body, including growth and development, metabolism, body temperature, and heart rate. The thyroid hormones are tyrosine-based hormones produced by the thyroid gland that are primarily responsible for the regulation of metabolism. Membrane receptors. Ligand-gated ion channel receptors, also commonly referred as iotropic receptors, are a group of transmembrane ion channel proteins which open to allow ions such as calcium, sodium, potassium to pass through the membrane in response to the binding of a chemical messenger, such as a neurotransmitter. Enzyme-coupled receptors, also known as catalytic receptor, is a transmembrane receptor where the binding of an extracellular ligand causes enzymatic activity on the intracellular side. Hence, a catalytic receptor is an integral membrane protein possessing both enzymatic, catalytic, and receptor functions. Lastly, G-protein coupled receptors are the largest and most diverse group of membrane receptors in eukaryotes. These cell surface receptors act like an inbox for messages in the form of light energy, peptides, lipids, sugars, and proteins. That concludes this presentation.